Biology 1107, presentation number 6, Energy and Enzymes. What is energy? Energy is the capacity to do work. That is a physics kind of definition, so let's figure out what work is. Work on one level is exerting force. That's what a physicist would say. Most folks would say that work is getting things done. There's all kinds of energy-intensive processes out there. And at the level of the cell, those energy-intensive processes could be making things, breaking things, transporting things, and transforming things. Every single one of those events could cost some energy. So energy is the ability or the capacity to perform work or get things done. Now, in biology, well, actually in the entire universe, energy takes two possible forms. One is called potential energy, and one is called kinetic energy. Very quickly, potential energy is energy at rest. If you're thinking about, for example, one of those old Roadrunner cartoons from way back in the day, which is like long before you were born, there was this guy named Wile E. Coyote who was trying to get the Roadrunner in the middle of the desert. And what he would do is perch giant boulders on the tops of mountains. And then he would push the boulder in the hopes that the boulder would crush the Roadrunner bird. But the fact of the matter is you got a boulder on top of a mountain. That boulder represents a certain amount of potential energy. That's stored energy. Sometimes potential energy is called stored energy. Sometimes it's called energy at rest. In biology, much energy is actually stored and contained in the form of food molecules. So we take our look at our molecule of glucose at right. And on our heart of hearts, we understand that glucose is constructed of carbons, hydrogens, and oxygens, all covalently bonded. And those covalent bonds represent shared, energized electrons. So that glucose molecule is jam-packed full of energy. Now, on the other hand, if the energy's not potential, it's got to be kinetic. So what's kinetic, Dr. Knaus? Well, kinetic is energy being expended. Now, be careful. We're not talking about energy being lost. We're talking about energy being used. We're talking about energy being converted from a storage form to one where it's actually doing something. That's kinetic energy. So in contrast to potential energy, which is essentially energy in some form that could be maintained almost indefinitely, kinetic energy is here and gone. Now, when I say here and gone, I'm going to understand that when energy does work, it is not lost. It is not destroyed. It is converted, though, into something that is far less usable. Hold that thought. We're going to talk about the laws of thermodynamics, and that will help explain that. The laws of thermodynamics are the laws that govern energy in the universe. And there's two of them. The first is the law of conservation of energy. It's called the first law of thermodynamics. It states energy can neither be created nor destroyed. So, relevance to the universe, we understand that the universe was created perhaps out of the Big Bang, but the fact of the matter is the amount of energy that we find in the universe right now is the same amount of energy that was found in the universe very moment it was created. And the amount of energy contained in the universe when it all ends will be exactly as it is now. So energy is neither created nor is it destroyed. Relevance to biological systems, the amount of energy you find in you or the amount of energy I see in my cat at a given moment represents the full amount of energy that is contained in the molecules of the cat and perhaps in the food the cat just ate. Well, I could take that cat and have it walk around the block, and it can come back, and I know it has expended energy in doing that. Yet, the amount of energy that is represented by that cat and its walk around the block is exactly the same as it was before he took the walk. Well, how's that, Knaus? I mean, he's expended the energy, right? Well, no, because the fact of the matter is when he did expend the energy, that energy was converted into heat. That energy propelled him around the block, but as it was expended, as it was converted to potential, to kinetic, and then expended, it was converted into heat, which is a far, less usable form. And for most of us, it's dissipated into the atmosphere, but it's still there. So the laws of thermodynamics have relevance to the universe. They have relevance to individual organisms. And the thing is when we think about the cat walking around the block, that energy, which we started as potential energy, became transformed to kinetic energy. And technically, the expending of that energy for the cat to walk around the block was a transformation as well. So energy transformations kind of work hand in hand with the first law of thermodynamics, because we understand, yes, energy cannot be created nor destroyed, but we might get the impression that it is, but we need to understand it's probably those transformations that are giving us the idea that perhaps the energy is being destroyed when it's really only being transformed into a far less usable form. But when it's said and done, it's the same amount of energy, just a little bit different form. Let's take a look at the second law. The second law of thermodynamics is sometimes called the law of entropy, and the word entropy means disorder. This is also going to go hand in hand with the first law, because the second law understands that when you have an energy transformation, that energy transformation is never 100% efficient, which means energy will always be degraded into some less usable form. Relevance to the universe? Well, let's see. Physicists will tell me that when the universe was created out of the Big Bang event, the universe is now constantly expanding. We look at the universe with telescopes and things and understand that the universe is constantly expanding. See, if the universe was created out of a Big Bang, that would have been a highly concentrated amount of energy yielding the universe. And as the universe is moving outwards, that is technically a form of entropy, of disorder. Relevance to biological systems, let me talk about that cat again. The cat walked around the block. Well, in walking around the block, the cat took advantage of some stored energy, potential energy, converted into kinetic energy, which was then transformed into heat radiating from the cat, and probably also some friction, at least involved in the contraction of the cat's muscles and maybe in the action of the cat's toes on the pavement. But regardless, there's this energy transformation going on, and that energy transformation is seen a transformation from a high quality form of energy in the form of that stored energy to a low quality form of energy in the form of heat. So those are energy transformations. The bullet point down under second law, it says energy transformations. Please write down energy transformations are never 100% efficient. And that's true even non-biological systems. When you go and sit in your car and you turn it on and you drive 100, 200, 300 miles, your gas gauge goes down. All right, well, that gas represents a high-quality form of energy, but when you're driving along the road, you're losing energy in the form of friction, which is stress of the tires on the road, generates heat. You got heat coming off the car, so technically, the operation of your car is an example of an energy transformation, and it is not 100% efficient. But the bottom line is, in the universe, in biological systems specifically, when you do an energy transformation, and whether that means making the cat walk around the block, or keeping the cat alive, or transforming things in that cat's cells, maybe making things, maybe breaking things, all of those energy-intensive transformations convert potential energy to kinetic energy, and kinetic energy expended in whatever activity is would then be transformed to heat, which is the lowest quality form of energy. Make sure you note that on the last bullet where it says quality of energy, heat, lowest quality form. It probably goes without saying that energy is critical to life. And it's going to be critical in the sense that it is going to be a requirement of some reactions and perhaps a product of others. So let's take a look at our first two bullet points here. We're going to identify reactions based on the role of energy in those reactions. There are endergonic reactions, which are energy-consuming reactions. You will see here reactant A plus energy yields product B. That's an energy-consuming reaction. That's an endergonic reaction. And then we have exergonic reactions. B yields A plus energy. Now, as a rule, endergonic reactions and exergonic reactions are coupled. You cannot have one without the other. And the reason is because of that law of conservation of energy. Energy can neither be created nor destroyed. And so whenever you have a reaction, there is going to be some place that energy has to go or some place where that energy comes from. So let's take an example. I see here hydrolysis of ATP, which is the breaking of ATP, our biological fuel molecule. It's called adenosine triphosphate. It contains a heck of a lot of stored energy. If we break that ATP molecule, process is called hydrolysis, we will release energy. So that is an exergonic reaction. So up above where you see the bullet point that says exergonic reaction, you might write down to the side somewhere, hydrolysis of ATP. You break ATP, you release energy. Now, on the other hand, going back to our example, we see we are pairing hydrolysis of ATP with muscle contraction. I have a coupled set of reactions. Muscle contraction is energy intensive. So muscle contraction is an endergonic reaction. So up at the very top bullet where it says endergonic, A plus energy yields B, I'd label that muscle contraction. So then when I take a look at the actual coupled reactions at the very bottom of the slide, I see the muscle contraction reaction, I see the hydrolysis of ATP reaction, and I have also included an arrow showing you that you cannot have one without the other, because if hydrolysis of ATP releases energy, that energy needs to go somewhere. Oh, we're going to use it in the contraction of the muscle. And if we're going to contract the muscle, where does that energy come from? Comes from the hydrolysis of ATP. Make sure you know what are endergonic reactions, what are exergonic reactions, and what we mean by coupled reactions. Now, there's one more thing I will mention here, because it is going to be relevant to something we will be talking about a little bit later in this presentation, and certainly when we get into presentations on cellular respiration and photosynthesis. And I'll tell you, I think I probably mentioned this earlier anyway, and that is this. An example of an endergonic reaction is photosynthesis. Because in photosynthesis, what we're doing is we're taking simple ingredients, carbon dioxide, water, and the sun's energy, and we're creating a molecule of glucose. That's an endergonic reaction. The sun's energy is being consumed in the production of the glucose molecule. Well, then we also talked about something called cellular respiration. And if we understand that photosynthesis is making a food molecule, then cellular respiration is breaking a food molecule. And cellular respiration is an exergonic reaction. So what I would do, and I know you're probably running out of room on your paper, but the fact of the matter is I would make a note that an endergonic reaction is photosynthesis and an exergonic reaction is cellular respiration. Two awesome examples of energy reactions. And then obviously they do kind of fit together, because I mean, if you're going to be making a food molecule, you can break a food molecule as well. And that's happening within the plant's cells. And even if you ate the plant and you didn't make the food molecules yourself, you're still carrying out an exergonic reaction that is ultimately linked to that event of photosynthesis that created the darn food molecule to begin with. I guess we better identify what are energy-containing molecules. Now, some of them are a no-brainer. So, for example, you'll look at our first bullet point here. Food molecules, carbohydrates, monosaccharides, disaccharides, polysaccharides, proteins, including polypeptides and amino acids, and lipids, including fats, oils, sterols, and phospholipids. All of those compounds contain energy stored in the form of energized electrons. They are energy-containing molecules. They represent potential energy. Then I see my second bullet point, and in my heart of hearts, I understand if I break down those food molecules, I can make ATP, which is my biological fuel molecule. No, it is not a carb. No, it's not a protein. No, it's not a lipid, but it is a source of energy. It is immediately usable when it's available, and it is universally usable when it's available. We'll talk more about that in a minute, but we're going to understand that, yes, food molecules are energy-containing molecules. ATP is an energy-containing molecule. And then I got one more bullet point, which is kind of unique and special. It's confusing to a lot of students because these are molecules that have a role to play in certain metabolic reactions. And they don't hang around very long, but they still contain energy, and they're still incredibly important. So what you might want to do is write down some things as I talk about this bullet point. First off, it says these are energy-containing reduced molecules. Now, underline the word reduced and make sure you understand these are molecules that have gained energized electrons. That's what the word reduced implies. They've gained electrons, and the assumption is if they're energy-containing, well, the electrons they've gained are energized. Now, the great thing about these molecules is they are technically managing those electrons. So, in addition to writing down the fact that reduced means gaining energized electrons in this case, you might also want to make a note that these molecules are energized electron managers. So, why would you need a manager for energized electrons? Well, here's the deal. I want you to realize that an energized electron is a highly reactive thing. And if it's a highly reactive thing, it's a potentially destructive thing. But on the other hand, it contains energy, and so while it might be destructive, if we are able to manage it, we might be able to use it for our own benefit. And that's what we do when we break down a food molecule. When we break down a food molecule, we release the energized electrons from that food molecule. But the thing is those energized electrons need to be managed until I get a chance to do something constructive with them. And that's what these reduced molecules are involved. For example, if you take a look at our bullet points, you will see that one of these energy-containing reduced molecules is called NADH plus H. It is important in cellular respiration. And right next to it is another molecule called FADH2, also important in cellular respiration. Those two molecules are very similar, and they do the exact same thing. And that is when you are breaking down a molecule of glucose, you're releasing those energized electrons. But what I want to do is I want to hold on to those energized electrons before they do something stupid, before they dissipate, before they react with something, before they destroy something. So I'm going to hold on to them until I have a chance to do something constructive with them. Hence, NADH plus H and FADH2 are energized electron managers in the process of cellular respiration. Turns out the same thing happens in photosynthesis. In photosynthesis, when the sun is shining on the plant, somehow what we got to do is we've got to capture that energy and stick it on some electrons. And when we put the energy on those electrons need to be managed until we can do something else with them. Well, smack dab in the middle of the process of photosynthesis, we are going to manage electrons in the form of a compound that we see here called NADPH. That's a cousin to NADH plus H. As a matter of fact, they're almost the same, with the exception of the presence of the phosphate group on the NADPH. But they do the exact same thing. In the process of photosynthesis, it becomes necessary for me to manage energized electrons. Now, one more thing, and that's this. We will not be talking about the detailed role that these compounds play until much later on in the semester. We'll have a presentation on cellular respiration. We will have a presentation on photosynthesis. And at that point, we'll do the details. So right now, all you need to know is NADH plus H, FADH2, NADPH are all energy-containing reduced molecules. They are energized electron managers. So they add into our list of energy-containing molecules found in all of biology. Food molecules, ATP, and energized electron managers. Well, I think it's about time we talk about ATP. We know that ATP is cellular fuel. It is the biological fuel molecule. Bacteria, algae, fungi, plants, animals, you name it, if it's alive, it will use ATP. We're going to talk about ATP in terms of how it is made and how it's broken. I'm going to use a schematic here. ATP is formed through a process called phosphorylation, which is nothing more than a $64,000 term for adding phosphate to something. So if you take a look at our bullet point, we're going to understand that this is actually an endergonic reaction. If we are going to make ATP, what we have to do is we're going to have to basically add energy to do so. Then on the other hand, there's a second reaction that we're going to call hydrolysis, which is breaking ATP. All right, well, let's take a look at the diagram. I see ATP labeled there at the top, schematically as A, P, P, and then a squiggle, and a third P. So here's the basic idea, is that ATP is adenosine triphosphate. So the A stands for the adenosine. I got 3 phosphates, but you'll notice the bond to the third phosphate is unique. That's because the bond to the third phosphate is commonly described as a high energy bond. That bond to the third phosphate is where we find this usable energy. This is where we find the energy that this molecule will contribute when some need arises. It's the fuel in the fuel molecule, so to speak. So now, if I want to get that energy out, what I'm going to do is I'm going to break that bond to the third phosphate. Hence, I see the arrow pointing down. It's labeled hydrolysis. Hydrolysis is a term commonly used for the breaking of bonds. So you break the bond, you release the energy, and then what you get down below is the energy released and the third phosphate all by itself, and then something that is portrayed there as A bond P bond P, which is adenosine diphosphate. Well, then what I can do is just go ahead and regenerate my ATP, which is basically go ahead and make ADP, add the phosphate, add a bunch of energy, follow the arrow pointing up, is called phosphorylation, and we get right back to our ATP molecule. Now, what I'm looking at here is commonly called the ATP cycle, and if I had my druthers when I made the slide, I would have labeled that at right. This is the ATP cycle. It is a cycle of making and breaking ATP. And so the idea is that if we can basically break the ATP, we can use that energy to perform cellular work. making things, breaking things, transporting things, and transforming things. I need to make a comment about this term phosphorylation. Most times you will associate that word with the making of ATP. Remember, phosphorylation literally means adding phosphate to something. But the word phosphorylation will also describe the activation of many proteins in the cell. Many times, if a protein is to do a job, it needs to get activated. And that activation involves kind of an injection of energy to the protein, and that is accomplished through the action of an enzyme called kinase. So you will see our bullet point says a kinase is an enzyme that will actually activate a protein. Take a look at the illustration below. You will see a deactivated protein, purple thing on the left. Follow the arrow. You'll see a protein kinase illustrated across the top, at which point you see that ATP is being converted to ADP. What we're going to do is we're going to take that third phosphate from the ATP, and it's a high-energy phosphate, and we're going to add it to our protein. Well, I see that over on the right-hand side of the illustration. There's my protein. Now it's got a high-energy phosphate bonded to it. It has been phosphorylated. And the arrow indicates to me that that particular protein at that point has been activated. Now, on the other hand, there are other enzymes called phosphatases that will deactivate proteins. Phosphatase would be an enzyme that removes a phosphate. Follow the arrow, protein phosphatase. Phosphate is removed, and I'm right back to my original deactivated protein. So make sure you're aware of the fact that phosphorylation, yes, is a term that is used to identify the making of ATP, but is also a term used to identify the activation of certain proteins. And that activation is a consequence of a high-energy phosphate ultimately derived from ATP to begin with. Now, I know I told you that we weren't going to go into detail on these reduced energy-containing molecules. You remember they were NADH plus H, FADH2, and NADPH. Well, I lied. I can't help it. I really need to discuss NADH plus H. We're going to use that as our focus. We're going to use that as our focus because we are going to be talking about cellular respiration in a lecture or two, and it's going to be critical that we have a good idea of how this particular compound works. So here's what I need you to know. It is a very common compound, and every biology student on the face of the planet, if you don't know about this, you will. It is a molecule that functions as a manager of energized electrons. NADH plus H goes by several different names. It depends on what textbook you're looking at. Depends how lazy the individual is. If you're not lazy like me, on a slide, you're going to write it as NADH plus H. Now, I'm going to give you a clue. The H's are kind of a sign that there are electrons on this molecule. The H's are a sign that electrons have been gained. Some folks, if they're lazy, will just write this as NADH, or sometimes they will write it as NADH2. It doesn't matter whether it's NADH plus H, NADH, or NADH plus 2. It is the same basic compound. It is a compound that receives energized electrons, and when it does, that is a process of reduction. NAD plus is what we start off with. That's the oxidized form of the molecule. We gain a couple of energized electrons, a couple of hydrogens come along for the ride, and you get the reduced energized form I see there on the right. It's the basic process, how it works. Now, those two electrons I said are energized. And once again, I will ask you to remember they're highly reactive and potentially destructive. It's one of the reasons why we need NADH in this case. It is an energy-containing reduced intermediate. That means it plays a role. It is an intermediate. It plays a role in cellular respiration. Well, let's see, Dr. Cass. That means we're breaking apart a food molecule. We're extracting the energized electrons. And you said those energized electrons, by virtue of being highly reactive, by virtue of being potentially destructive, we need to manage them until we can do something constructive with them. Well, the constructive thing is we can use those energized electrons as a way of making ATP. So when you start breaking down food molecules, you're extracting energized electrons. Those energized electrons will ultimately be processed to make ATP. Look at our last bullet. It says must be processed by electron transport chain. We're going to talk about that on our next slide. An electron transport chain is a series of molecules that are collected to process these energized electrons to extract the energy. In the bottom of that slide, after it says extract the energy, you write down and make ATP. And we're going to see this on our next slide. Let's take a look at what an electron transport chain is and what it does. And we're going to take a look at it in a first respect on this slide, which shows us kind of schematically what is involved. Then we'll actually take a more detailed look at it and how it actually exists in the context of a cell. Let's start out with a simplistic version first. Well, Dr. Canals, it doesn't look terribly simple to me. Well, let's see how simple it is. Take a look at the three balls on the far right-hand side of the slide, and that is an electron transport chain. An electron transport chain, I said, was a series of molecules that are designed to process electrons. Well, I see here they're kind of arranged in series, and they're sometimes called electron carriers. And they're called electron carriers because they easily gain and easily lose electrons. And I'm getting that impression looking at the little arrows there. So electron carriers are easily reduced and easily oxidized, and they're going to pass those energized electrons along. Now, the reason why those electron carriers are drawn going downhill is to indicate energy level. As the electrons go from the first to the second to the third energy carrier, they are losing energy. Well, you'll notice that the energy is coming off as a big fat yellow arrow, and we're ultimately going to use that energy to make ATP. The details we will see on our next slide. So, electron transport, what is it? Well, it is an important part of cellular respiration. It is a method by which energized electrons from NADH plus H are processed to extract their stored energy. It says we begin with the oxidation of NADH plus H. Well, Dr. Canales, I see NADH plus H over there. It's got a big explosion around it. Well, I suppose that would tell me that the NADH plus H is an energy-containing compound. And after all, you did tell me that NADH plus H was an energized electron manager. So it's managing energized electrons as I look at it. Follow the arrow. All of a sudden, the NADH plus H is converted to a unenergized form, NAD plus. Technically, that's the oxidized form of NADH plus H. It's lost those electrons. Oh, I see the electrons over there, Dr. Canals. The energized electrons have been removed from the NADH plus H molecule, and I'm following the arrow, and those energized electrons are going to head down the electron transport chain. The electrons go to the first carrier, to the second carrier, to the third carrier, and as they run down from carrier to carrier to carrier, the energy is removed and ATP is made. Well, there's one more thing here, and it's pretty darn important, and that is we're going to understand that electrons are matter and they cannot be destroyed, which means something has to be done with those electrons. They come down the electron transport chain. They cannot stay there at the very bottom of the electron chain. They literally gum up the works. So once the electron gets to the bottom of the chain, it jumps off the last electron carrier and hooks up with oxygen. Oxygen has a role to play in electron transport, and it's incredibly important. We're going to say that oxygen is the final electron acceptor. And I see that as the last bullet point on my bulleted list over there. Oxygen is there to remove that electron from the electron transport chain, which means once that electron is removed, other electrons can continue to run through the process. Well, Dr. Kness, what happens to the electron? Well, the electron hooks up with oxygen. A couple of hydrogens come along for the ride. I'd like to think of hydrogens as electron groupies. Every time you've got free electrons, there will always be hydrogens around. But look, oxygen plus a couple of electrons plus a couple of hydrogens gives me water as a product. Dr. Kness, does this have something to do with the fact that you and I breathe oxygen? That's a very astute observation, because the reason you and I do breathe oxygen is because that oxygen will be used as the final electron acceptor in the process of cellular respiration in all our cells, and that oxygen is key to our being able to process those electrons. So go back and look at the slide and make sure you understand what you're looking at, because we're going to take a look at a lot more, let's say, realistic or sophisticated view of it in our next slide. We're going to run through this again. NADH plus H, reduced energy-containing compound, is oxidized, converted to NAD plus. The energized electrons, which you remember are highly reactive and potentially destructive, that's why they were being managed by the NADH plus H molecule to begin with. They are now going to be passed down the electron transport chain for a constructive purpose. We're going to remove the energy. We're going to make ATP. When we are done, the electrons hook up with oxygen, the final electron acceptor, and they, along with the oxygen, along with a couple of hydrogens that come along with the ride, form a molecule of water. That's the general idea. Let's see how it actually happens in our cells. Well, you'll remember that the process of cellular respiration takes place in the mitochondria in our cells. Mitochondria are sometimes called the power plants of the cells because they crank out ATP. So on this slide, I see, in fact, on the second bullet point, it says something about a mitochondrial membrane. So off to the right-hand side in big capital letters, I want you to write down in mitochondrion. Electron transport occurs in the mitochondrion. So if I'm looking in the cell and I want to know where this processing of NADH plus H happens, it happens in the mitochondrion. And this is what it looks like. If you take a look at our illustration, I'll clue you in on the illustration first, and then we'll look at the bullet points. The horizontal line that is extending along from the left to the right, that is an inner mitochondrial membrane. That's a membrane inside a mitochondrion, and it's got all kinds of things in it that are going to be critical to our being able to process those electrons and make ATP. I also want you to notice there are 3 small cylinders embedded in that inner mitochondrial membrane. Each one is called a cytochrome. Those are our electron carriers. Here, I need you to know that officially, electron carriers in an electron transport chain are molecules called cytochromes. And they are easily gain electrons and they easily lose electrons. They play hot potato. I don't know if you ever heard of that game. The game of hot potato is the idea that you give someone a hot potato and they pass it on, pass it on. And obviously, if you drop the potato, you have to leave the chain. Kind of a stupid, silly game. But the fact of the matter is that's what's happening. We're passing these energized electrons from cytochrome to cytochrome. Just like we did so on the previous slide. But now you'll remember on the previous slide, our electron carriers were illustrated going downhill. Now what we're doing is we're illustrating them more realistically as how they physically occur in a membrane. But they'll do the same thing. So I've got my series of electron carriers. I'm going to call them cytochromes. They're easily reduced. They're easily oxidized. Take a look at the far left. There's my 2 energized electrons from NADH plus H. We're going to pass them from cytochrome 1 to cytochrome 2, cytochrome 3, and then they're going to jump probably to our final electron acceptor. But now this process, what we're about to look at, is going to explain to us how we actually make the ATP. In the previous slide, there was nothing more than a big fat yellow arrow showing us that the energy pulled off of those electrons made ATP. That's not good enough. We need to get into the details. The details involves a process called chemiosmosis. We will define that as we look at our next slide. Here's our next slide. The details on how we are going to make ATP from energized electrons in a process known as chemiosmosis. Now, the word chemiosmosis implies osmosis, which is kind of like the movement of water molecules across a membrane. But in this case, we are not moving water molecules across the membrane. We're going to move hydrogens across the membrane. So chemiosmosis is going to involve a proton gradient. Somewhere next to that word, chemiosmosis, write down proton gradient makes ATP. Chemiosmosis, right next to that word, proton gradient makes ATP. So what I'm going to do is I'm going to run you through a play-by-play of the illustration. Then we'll talk about the bullet points. But if I run you through the illustration, the bullet points will make sense. So we're going to keep in mind we're starting off with energized electrons from NADH plus H. We need to manage those energized electrons, at least manage them constructively, so we can make some ATP. And this is going to happen in the mitochondrion. So there's our inner mitochondrial membrane, and there is our electron transport chain, consisting of 3 cytochromes. We take our energized electrons, far left. The energized electrons get passed from the first cytochrome to the second cytochrome to the third. The energy on those electrons are going to be used to pump protons from one side of the membrane to the other. So you will notice that each of those cytochromes features a little fat purple arrow pointing upwards. And it's indicating that I am moving hydrogen ions from one side of the membrane to the other. As a matter of fact, I see there is now a gradient of hydrogen ions. Now, you remember hydrogen ions are sometimes called protons. So I've got a bunch of H pluses. I got a bunch of protons above the membrane and very few below. That's a proton gradient. Matter of fact, I can go ahead and look at my first two bullet points. I can't resist. Energy from the electrons is used to pump protons across the inner mitochondrial membrane. It creates a proton gradient. Go back to the illustration. Off to the far right-hand side, you will see a unique cylinder there that is labeled ATP synthase. That is an enzyme. And it's an enzyme that operates when protons flow through it. I like to think of ATP synthase as something like a turbine in a hydroelectric dam. If the hydrogens flow through the ATP synthase, and I'm looking at the illustration right now, I notice I'm going to be making ATP. In a hydroelectric dam, water is going to flow through a turbine, and when it does so, the turbine turns and cranks out electricity. It's the same basic idea. So let's go ahead and go back and look at our bullet points. Energy from the electrons is used to pump protons across the inner mitochondrial membrane. That creates a proton gradient. The protons are desperate to flow from a high concentration to a low concentration, which means my protons flow back across the membrane through ATP synthase. As they flow through ATP synthase, ATP is produced. And if you go back and look at the third cytochrome, that you realize, of course, that when the last couple of electrons jumps off, they are going to be picked up by oxygen, which is the final electron acceptor, and we make water as a consequence. Make sure that you know that chemiosmosis is a process by which we make ATP using a proton gradient. And make sure that you know that that proton gradient is created by those energized electrons that we obtained from our energized electron manager, NADH plus H. Well, how about that? Here's my hydroelectric dam analogy. See, we're making ATP, and that's an event of phosphorylation. ATP synthase is like a turbine in a hydroelectric dam. The protons are like the water behind the dam. ATP is like the electricity produced as the water flows through the turbine in the dam. Now, one little bit here. If I obtain energized electrons from NADH plus H, those energized electrons in one NADH will yield 3 ATP of energy. On the other hand, if those electrons come from a molecule called FADH2, and you remember that's like a cousin of NADH, I'd have to say it's like a poor cousin because it doesn't contain as much energy. The electrons from one molecule, FADH2, will yield 2 ATP, not 3. This is going to make more sense when we get into the presentation on cellular respiration. So once again, make sure that you are aware of how this works. It's like a hydroelectric dam. We're cranking out ATP through a process of phosphorylation. Using protons, it's called chemiosmosis. And in doing so, one NADH will yield 3 ATP of energy, and one FADH2 will yield 2 ATP of energy. Okay, we've covered the notion of energy, what are endergonic and what are exergonic reactions, ATP, the idea of energy-containing molecules, in particular energy-containing intermediates, energy-containing reduced molecules, and how they're actually processed through electron transport as application to the process of cellular respiration. Things that we're going to cover later on. But right now, let's turn our attention to a whole nother topic that will actually wrap up this particular presentation. We need to talk about enzymes. And we've probably mentioned enzymes before. Enzymes are molecules that initiate and accelerate biochemical reactions. They're sometimes described as catalysts. A catalyst is something that starts something. So, definition: enzymes, molecules that function as biological catalysts, molecules that initiate and accelerate biochemical reactions. Now, in particular, those reactions, as a rule, are energetically improbable. Now, what do you mean by energetically improbable, Canals? I mean, these are reactions that just cost way too much energy. As a matter of fact, if you were to tally up all the reactions that keep you alive, reactions that occur in your individual cells, you couldn't possibly consume enough energy, calories, to support those reactions. They're just too expensive. So life is, in a nutshell, energetically expensive. But somehow we manage to stay alive. Somehow we manage to carry out these reactions. And the thing that makes that all possible are these molecules called enzymes. Because we're going to see in a minute that enzymes actually give us an energy discount in making a reaction possible. So let's take bullet point number 3, and then we're going to take a look at the graph to the immediate right. Any reaction is going to require something called activation energy. Now, activation energy is sometimes abbreviated capital E subscript A. Stands for activation energy, or sometimes energy of activation. It is the amount of energy that must be present for a reaction to proceed on its own. So hence, I'm looking at the graph, and I notice the graph shows me a reaction. I've got reactants, A plus B, on the left. I have products, C plus D, on the right. That would be my reaction. A plus B yields C plus D. And if I take a look at the blue line is indicating to me the amount of energy that is present when that reaction occurs. I notice that the energy of A and B are relatively low, but then I notice I have to supply a significant amount of energy in the form of that really, really tall hill represented by the upward curve of the dark blue line. That is activation energy. And you see it's bracketed as E sub A over on the right. That means I need to supply that much activation energy to get to my products. That's expensive. That's energetically improbable. But take a look at this. There's another line on this graph. It's in red. If I supply an enzyme, that enzyme gives me a discount. Enzymes lower activation energy. Please write that down somewhere near this slide. Enzymes lower activation energy. They provide us a discount. And so I see the red line makes a little hill as well, but it's nowhere near as tall as the dark blue one. So I'm lowering activation energy in the presence of an enzyme. And at that point, what was an energetically improbable reaction then becomes probable. Okay. Now, there's one more thing I should mention about enzymes before I move on to my next bullet point, and that is this. For the most part, and I would have to say 99.9% of enzymes are proteins. See, proteins are really big molecules, and we said once upon a time they have very specific shape, and that very specific shape is really convenient for binding to the reactants and triggering this reaction to happen. So enzymes, as a rule, being molecules that function as biological catalysts, being molecules that lower activation energy, enzymes, as a rule, are proteins. And they bind to the reactants. Now, moving to bullet number 4. In an enzyme-mediated reaction, and sometimes they're called enzymatic reactions, the reactants are commonly called substrates. And the reason they're called substrates is because they will actually bind to the enzyme. You take a look at my simplistic illustration, you'll see an enzyme, looks like Pac-Man. You'll see a wedge-shaped opening. That is what's called the active site. And the active site is the place on the enzyme. It is a physical, three-dimensional place on the protein where the reactants or the substrate are going to bind. So in this case, in our example, we just got one substrate binding to that enzyme, but it could be several. For example, my reaction up there, A plus B. 2 substrates could in fact bind to the enzyme. So when you get this combination of the two, and it's a very specific relationship. As a matter of fact, 5th bullet mentions enzyme substrate specificity, means that any given enzyme really is only going to be able to bind with and interact with a specific substrate. Case in point, in your saliva, there is an enzyme called salivary amylase. Salivary amylase has a job to do. Its job is to break apart starch molecules. It doesn't break apart proteins. It doesn't break apart fats. It doesn't break apart fiber. The only thing it works on is starch. So the relationship with amylase and starch is one of enzyme substrate specificity. There are some fungi that are able to break down wood. They have an enzyme called cellulase. It allows them to break down cellulose in cell walls. Now, cellulase will only work on cellulose. It won't work on starch. It won't work on anything but cellulose. Enzyme, substrate, specificity. And that all boils down to the shape of the active site. The shape of the active site fits the substrate, allows the combination of enzyme and substrate to occur. So I'm looking at my lower right illustration, and I see, yes, there is a substrate that seems to be very tightly bound to my enzyme at the active site. The combination of substrate and enzyme is called the enzyme-substrate complex. It is an absolutely critical part of the enzyme-substrate reaction. So let me talk about this, and I'm going to ask you to write down 3 things. The enzyme-substrate complex. Number 1, it is a perfect fit between the enzyme and the substrate. Number 1, it is a perfect fit between the enzyme and substrate. Number 2, it is temporary. It lasts only as long as it is needed. Number 2, it is temporary. And number 3, perhaps the most important one, is the enzyme-substrate complex is where activation energy is lowered. Number 3, it is where the activation energy is lowered. It's by virtue of the fit between the substrate and the enzyme that creates that discount that makes that reaction possible. Now the deal is once the reaction is done, There is no need to maintain the enzyme substrate complex. So once I'm done processing the substrate, the whole thing will come apart. And what I've got is I've got enzyme and products. Well, what about that enzyme, Dr. Knaus? Well, if you take a look at the next bullet point, after enzyme substrate complex, I see enzyme unchanged. Technically, after involving interaction between an enzyme and substrate, the enzyme, in fact, is unchanged. However, got to make a comment about this. Theoretically, yes. On a theoretical level of things, yes, the enzyme is unchanged, which means, theoretically, the enzyme could interact with other substrate molecules over and over and over again. I mean, technically, one enzyme molecule could, theoretically, engage in millions of reactions and always deliver the same results. However, realistically, things are a bit different. So there's two concepts behind the enzyme after the enzyme substrate complex is completed. I got the idea the enzyme is unchanged, at least as a theoretical idea, but then I've got to talk about the realistic. What's the realistic, Knaus? The realistic is this. If an enzyme engages in a reaction, it is going to experience wear and tear. It's like buying a car. You buy a car and you know you drive off that lot in your heart of hearts, that $30,000 automobile, if you're so blessed. In your heart of hearts, that $30,000 automobile a year from now will not be the same car you purchased. And Lord knows, I mean, the Pontiac van that I purchased way back in 2001, 12 years later, was not the same car. I mean, it was a gorgeous van when I bought it, but for crying out loud, 12 years later, it had 275,000 miles on it. The windows didn't work. The car didn't get much beyond 45 miles an hour when it started to shake. It was miserable. It was well-deserved. I mean, the darn car just wore out. It was experiencing wear and tear because it is a physical, mechanical thing, and the same thing happens with that enzyme. It is a physical, mechanical thing because it does have a mechanical aspect when it binds to that substrate and triggers whatever reaction needs to be triggered. So, whereas theoretically enzymes are unchanged, realistically they do experience some wear and tear. Got to acknowledge the possibility does exist that an enzyme can actually work several times over and over and over again, but at some point it's going to experience that wear and tear. And at some point it won't work as well as it did. Okay, I mentioned the fact that there is this perfect fit between the enzyme and the substrate. Now let's talk a little bit about that. The way that fit develops is explained by something called the induced fit model. The idea is that the enzyme's active site is not necessarily perfect initially for that substrate. You take a look at our illustration to the right, you will see that the active site of the enzyme, that gap, is much too wide for the substrate. It's not a perfect fit between the two. It's not like a hand and glove kind of thing. But I will tell you this, scientists have found that when a substrate does bind to an enzyme, there is a change in the shape of the enzyme that induces the enzyme to have a perfect fit with the substrate. Hence, the fit of the active site changes to make the fit perfect. That's what's meant by induced fit model. Finally, going to wrap up this particular slide by saying that, as far as enzymatic reactions go, we got two general types. There are synthetic enzymatic reactions where things are made, and there are degradative enzymatic reactions where things are broken down. So, for example, in the case of my graph up there, A plus B yields C plus D, that technically is a synthetic enzymatic reaction. On the other hand, if I was to modify my illustration below and have that green substrate break apart into two triangular substrates, maybe one red and maybe one blue, that would be an example of a degradative enzymatic reaction. What kind of factors affect how an enzyme does its job? Well, an enzyme's job can be thought of as enzyme activity. Enzyme activity would be how much product does an enzyme get me? What's the potential? Well, there's a number of different factors that can influence an enzyme's activity. And a lot of that boils down to the simple fact that the enzyme is a protein. And you'll remember it wasn't too long ago when we talked about proteins who said that proteins could be denatured. Proteins were big molecules. All right, well, I see here that pH can have an effect on enzyme activity. If the pH goes down, or conversely, the pH goes up, that could conceivably have an influence on the shape of the enzyme. Remember, a negative change in protein structure is denaturation. So technically, an enzyme can become denatured. It won't work. Or we could, in fact, maybe finesse it enough so that if I change the pH just a little bit, it might not work as well. And then if I change the pH just a little bit, maybe I could bring it back. Well, there's a second function here, a second factor, and the second factor is temperature. Temperature has an influence on random molecular movement. Something we mentioned not too long ago. We talked about the fact that molecules do move, random kinetic movement of molecules, and it's ultimately influenced by temperature. As temperature goes up, those molecules move faster and faster and faster. Now, in the case of substrate and enzyme molecules, if they exist in solution, as they generally do, if you increase temperature, you're going to increase the rate of molecular collisions. The more frequent the molecular collisions, the greater the number of reactions. So increasing temperature can increase frequency of molecular collisions. Therefore, increasing temperature can increase enzyme activity. Unless the temperature goes up too much. So what we're going to do is understand that the idea of how temperature affects enzyme activity is like a double-edged sword. One edge will increase enzyme activity due to increasing temperature, but on the other hand, if temperature goes too high, you can trigger a denaturation event, which would negatively impact enzyme activity. Well, my third bullet talks about the concentration of enzyme. If I have a higher concentration of enzyme molecules, that means I have a higher number of active sites. The more active sites I got, the more reactions possible. So, yes, concentration of enzyme can have an impact on enzyme activity. On the other hand, what about substrate concentration? Well, if the number of substrate molecules increases, now it kind of depends on the number of enzyme molecules you got, but let's suppose I've got 100 enzyme molecules, and that means I got 100 active sites. And I got 100 substrate molecules, that means I got one molecule per active site. And that's about as fast as I can go. That's my maximum enzyme activity. If I increase the number of substrate molecules to 1,000, and I still only have 100 enzyme molecules, I'm still going to have the same reaction rate. because I can only occupy 100 active sites at any given time. So it's not the concentration of the substrate that makes the difference. It's the concentration of the enzyme that makes the difference. All right, well, I got one more bullet point here, or two more actually. We're going to talk about cofactors and coenzymes, and then we'll talk about inhibitors and activators. As a matter of fact, I think I'm going to have a slide about inhibitors and activators. So let's just talk about cofactors and coenzymes right now, and then we'll shift to the next slide. Some enzymes, folks, are not capable of working on their own. Some enzyme molecules need a little help, and that help is in the presence of, or in the form rather, of cofactors and coenzymes. What are these canals? All right, a cofactor, make sure you write this down, is an inorganic atom that promotes an enzyme's activity. For example, there is an enzyme that has a role to play in reproductive development of humans. And that enzyme has a requirement for zinc. If zinc is not present, then the individual will not mature sexually. And that is a problem in some populations on the face of the planet where zinc deficiency exists. So zinc is an example of a cofactor, an inorganic atom that makes an enzyme's work possible. Now, many of the enzymes that are responsible for taking food molecules and converting them into ATP, many of the enzymes involved in cellular respiration, and there are a lot of them, many of those enzymes require vitamin B compounds in order to do their job. Now, vitamin B is an organic compound. It takes a lot of different forms, but vitamin B, in this case, is an example of a coenzyme. If a cofactor is an inorganic atom that makes an enzyme's job possible, a coenzyme is an organic molecule that makes an enzyme's job possible. In the case of cellular respiration, the B vitamins are critical to helping those enzymes do their basic job. Make sure you know what are cofactors, what are coenzymes, and definitely make sure you know how those different factors of pH and temperature and concentration affect enzyme activity. The last bullet point on our previous slide mentioned inhibitors and activators. And very simply, I'm just going to say that activators are molecules that help to, as would indicate, activate the function of an enzyme. And they don't have to be coenzymes, they don't have to be cofactors, it's just a molecule present to trigger the action of an enzyme. Now, on the other hand, there are molecules that can inhibit the action of an enzyme. And this particular slide is going to focus more on those. We're going to talk about three basic types of inhibition. Now, an inhibitor is any molecule that is going to propose an ability to us to regulate an enzyme. If an enzyme is a protein, all I have to do is affect the shape, or specifically affect the shape of the active site, and I can regulate the enzyme from that standpoint. We're going to talk about three different types of inhibition. The first is called competitive inhibition. Then we'll talk about non-competitive inhibition, also called allosteric inhibition, and then feedback or product inhibition. First off, competitive inhibition. The word competitive is going to imply something competes for something, and in this case, we've got an inhibitor molecule that is going to compete for the active site with the substrate. Obviously, from my illustration there, if the inhibitor molecule is bound to the active site, no further reaction is possible. So an inhibitor that is a competitive inhibitor is competing for the active site with the substrate, and as a consequence can have negative impact on the enzyme's activity. Non-competitive inhibition is a situation where the inhibitor molecule binds to a completely different location on the enzyme's surface. The enzyme's really big, so there's plenty of places where you could conceivably establish some other binding event. So if I'm looking at my illustration towards the bottom of the slide, I see my purple molecule, the inhibitor, binding to the enzyme in yellow at some other place. And when it does effect a change on our active site. So the lower illustration shows me an active site that is rendered inactive because of this inhibitor binding at some other completely different place. Now that is sometimes also called allosteric inhibition. So definitely make sure you connect that name, allosteric inhibition, with non-competitive inhibition. They are essentially one and the same. Our last bullet documents something called feedback inhibition, also known as product inhibition. And we will be looking at the illustration at the lower left. Many enzymatic reactions occur as part of a greater whole. In other words, we might have a desired outcome. I'm starting off with substance A. I really want to get to substance C. But the thing is that desired outcome is not going to require just one reaction. It might require two. It might require three. It might require four. I've got a series of reactions that are going to get me to my desired endpoint. Every single one of those steps might involve a certain enzyme. So how could I regulate the whole shooting match? Well, probably the most logical way of doing that is to take my product and use it to feed back on the initial step in reaction. So I'm looking at this illustration here and I see substrate A is converted into substance B, which is converted into substance C, and the transition between A and B is moderated by an enzyme, and the transition between B and C is moderated by an enzyme. But the question is if I really wanted to regulate the production of C, how would I do that? Well, why don't I go ahead and use C, my product, as my mechanism for regulating. For the sake of argument, if I'm making too much substance C, well, gee, it's just going to feed back. It's going to go back to the very step in the process and maybe inhibit the enzyme that I started off with. So the feedback of substance C inhibits the production of B, which ultimately is going to inhibit the production of C overall. And the beauty of this situation is that if I get too much product, that product shuts down on the process. If then I don't have enough product, I'm opening the process right back up again. So it's the amount of product that's actually ultimately going to determine whether the process, the enzymatic process, works or not. That is called feedback inhibition, sometimes called product inhibition, in the sense that whatever molecule is being made has an influence on the entire enzymatic process.